I'm very happy to introduce our next speaker, Professor Peter Biggle from UC Berkeley. Peter holds honorary doctorates from the Hebrew University and ETH. He's a MacArthur Fellow, he's won the Cobb's Prize, he's a member of the American Academy of Arts and Sciences and the National Academy of Sciences, and he has a very wide range of research interests, including robust statistics, semi-parametric and non-parametric statistics, bootstrap, computational biology, and networks.
Thank you. I wander around a lot. I'm honored and grateful to be, it's a pleasure to be invited to this attempt to make statistics viable at MIT. I was instructed to have a talk which has some challenges in it, and also one which does not stress the mathematics. And I tried to do that. And what I'm going to do is I'm going to wander a bit. I'm going to talk about my view of statistics, and there'll be a little bit of math here and there, but not too much. Okay, so, it works. All right, so here's the topics. I'm going to talk about, again, what I view as the nature of statistics, the advent of big data and its consequences, and then a little bit of mathematics showing that a rather classic area of statistics can be seriously impacted by the fact that we're working in high-dimensional space. And then I'll talk about networks and examples, uses, and challenges. Okay, so, well, first, the trivial observation that statistics is about aggregates. And a single data point is of no interest, but of course, what a data point is not necessarily clear, right? A single genome is 3 × 10⁹ base pairs plus a lot of other stuff. And the other element that I feel is critical is that distinguishes statistics from other fields related to data, which is about everything, is that we try to think in terms of probabilistic models, or at least think probabilistically. We tell ourselves stories, and we may tell ourselves stories in the way of priors, in the way of models, but there has to be some probability theory there. Okay, and another way, at least I find, of thinking about statistics is, as I said, as a transfer science. That is, we transfer methodology for data analysis. So methodologies arise in all sorts of fields, and some of them are specialized. Some of them expire after a while. But if something turns out to be useful in a bunch of fields, it somehow, hopefully, enters the canon of statistics and is studied in that context in a general fashion. So some aspects, description is certainly an intrinsic part of statistics, and box plots are an example of methodology that's spread. Exploration, well, clustering is sort of the paradigmatic example of exploration of data. We're always looking for things which have something in common or are different from the others. Prediction, which is a large part of machine learning. Decision theory, which the last speaker referred to every now and then, not in that form, but the fact that you want metrics, you want loss functions, you want to have some measure of how well you're doing. And then validation, which is hypothesis testing, confidence regions, and finally models, and of course the classical and simplest example, which is Gaussian linear models. I want to talk very briefly first about regression analysis, but as an example of how the transfer goes on, but also later as an example of a very classical methodology which turns out to have some surprises in high dimensions. So formally, you're given observations, xi, yi. We typically think of y as being one-dimensional, but it doesn't have to be. And we want to determine the average behavior of y given x in some sense, and then want to put error bounds on aspects of that. Well, the history, and here I have Steve, who will undoubtedly point out that I've made some mistakes, but this is clearly, I think, an example of transfer. The first instances are Legendre in 1805 and Gauss, where they viewed these as ways of combining astronomical measurements. Then 1885, Galton in biology. I'm actually pointing to peaks that I know of. I know that there are all sorts of other points on the way. Then to the social sciences through the work of Carl Pearson and Udny Yule. And finally, with R.A. Fisher, to some extent, I guess, returning to the Gaussian model, and initially agriculture and then generally. There's an amusing note I can't resist making, which I believe is again, I may be corrected, which is that Gauss, it seems, initially, I mean, the initial method that was proposed was, not by Gauss, was least absolute deviations. But Gauss then proposed the method of least squares and justified, to some extent, the Gaussian distribution as that distribution such that maximum likelihood gave least squares. All right, so what's happened with regression analysis as time has gone on? First of all, it was implicitly about the conditional expectation of y, which is assumed to be linear in terms of x, and then the Gaussian model closely tied to that, because that is the multivariate Gaussian, is an example of that situation. Then we've had extensions, nonlinear regression, non-parametric regression, and then as Roger Kunker was referred to, we've started to think about other measures of centrality, right? Not just the conditional expectation given x, but the median given x, which is least absolute deviations. Here's you'll see I'll wander around a bit. So what this is just, again, showing you how much this technique has been transferred. So what this is, it's a plot of research areas ranging from geography to public, oh God, I can't even read it. There's no metric. This is simply a list of the top 100 from the Science Citation Index, the top 100 fields with the number of citations to regression analysis in each field from 1900 on.
That's probably an occupation that helps.
Thank you. You know, for some reason, I was looking for this. Now I see it. I was wondering why I didn't, okay. So of course I couldn't read it here either. But let's go on. All right, so now we have the advent of big data. And I'm actually starting not with the bigness, but with its complexity. I think really big data per se wouldn't have done anything, right? I mean, in fact, the physicists have always said, well, you know, if you have enough data, you don't need statisticians, you just take the mean, or whatever. But unfortunately, it's not only been big data, but very complex data. And here's something that I have been involved in, something called the ENCODE project. ENCODE stands for the Encyclopedia of DNA. And it's a project which started in 2003 and is continuing on to the present, and may continue on for another year or two. But the initial goal was to functionally annotate every element of the human genome. this was somewhat bold. It didn't happen, but what did come out is how very, very complex things are. And this came out as a consequence of, here's the work of the ENCODE project in terms of data. So it is big data, really. So 3,300 experiments, 1,700 different assays, 387 different cell types. And you multiply those numbers and you get something fairly large. And that is now, these things are now stored in the Santa Cruz genome browser and are the data which basically has led to an initial understanding of the very complex mechanism by which action is initiated in the cell. I mean, you have the genome, but then at first one thought, well, you have the genome, you have the letters, you have the genes, which of course have to be recognized, and then that's more or less the story. Well, the story is of course extremely complicated. There are promoters. Oops, what happened? Oh, this doesn't seem to. Okay, there are promoters in front of genes. The genes produce many transcripts. There are genes which produce as many as 10,000 transcripts. Not many, but there are some. Each transcript, of course, in principle produces a protein. So here you have one gene which produces that many, and there's tens of thousands of genes, so you can again imagine. And then very, very clever assays of different kinds are continuously being invented, which give you information about different regulatory regions. So now there's a big dispute. If you may remember that some years ago, people talked only about junk DNA, right? I mean, there were the genes, which are something like 15% of the genome, and then there was junk DNA. Well, if at least what the ENCODE project found was that almost all of what was left was biochemically active, not necessarily functional by any means, but that there was biochemical action. And why? Well, all sorts of stuff to tell the genes when to fire, whom to access, destroy bad stuff, etc. And this is all sort of contained in the genome. And so that's the challenge of big data. It's not only large, but it's complex, and the pieces you're interested in may be quite small. So here's more new issues. There's aspects which are viewed, at least initially, as being outside the statistical purview, but which are very important. Storage, data structures, computer architecture, speed of computation, all of this interacts with the procedures we focus on. And although it's outside of the purview of statistics, in fact, and I'm not speaking of myself, but I'm speaking of future generations, statisticians should have working knowledge of these issues. And as well as, and this is something that I feel is very important, which was also stressed, I think by Patrick, which is not in the field of science, but knowledge at the communication level to work with subject matter collaborators. In some sense, big data or complex data, high-dimensional data, has made that even more necessary, right? Because there are so many ways to go. And without some guidance, you're in trouble. You get nowhere. That's been very clear in, say, my collaboration with the people at ENCODE. Now, statistical issues. Well, probability models for RP, P of the order of or larger than N, and then complex structures. There are matrices, there are trees, there are manifolds. As I'll indicate at the end, networks and covariates can be thought of as tensors. You have the issue of validation, now that you actually are considering many, many hypotheses at the same time. And dimension reduction, which is in some sense critical. It's sort of vague. It doesn't just mean that you reduce go from R50 to R10 or something like that. But you change the structure of the data in such a way that you can try to understand aspects. And regularization, which is really part of dimension reduction. You penalize. Or, if you want to be a Bayesian, you put on priors. Except that you're not limiting yourself to priors which somehow reflect exactly what you think. Okay, now I'm switching. I want to show you now how, in this context of high dimension, there are curious phenomena which start to happen with our familiar, perhaps the most familiar model in statistics now, which is regression.
Linear.
Sorry?
Linear.
Linear, that's right, exactly. And so, okay, so this assumed model, here I'm going to use notation which I hope is sufficiently standard. So you have, this is actually regression with a random carrier, right? So that you can talk, but that just makes it easier. It's not, it's the same kind of phenomena can be thought of, but with more complexity and more difficulty when you have fixed designs. So you have the assumed model that you have independent observations, that y is, I don't know why this laser pointer seems not to work. Is it just because I'm not pointing correctly?
There's another one, there's a green one over there somewhere. It looks like a pen.
Yes, I see it. Thank you. And how does that, oops, there's a button, I guess. Oh, I see it, okay. Ah, excellent. And so you assume that y is a linear combination of unknown coefficients, but of columns of z. And then you have a deviation, if you want, an error vector, which is thought of as being independent of z. And this is used as an approximation to a more general model, which is also common in econometrics, which is y is mu of z, where mu doesn't have to be linear, and where all you have to say about the deviation is that its conditional expectation is 0. Oops, now I need the other one. Yeah, that's right. All right, this is awful, but it's sort of the basic theorem that you all actually know, except it's written in this way. So suppose that you do least squares, which is that, and you look at, we can have the explicit solution for the least squares estimate of beta, which can be written in this way. Then you can form the empirical covariance matrix of the z's up to scaling. And then there's the basic result which we use, which is in fact exact if everything is Gaussian, which is that if the expected value of ZZ transpose, the covariance matrix, is non-singular, beta 0 is true, the least squares estimate has fluctuations of order 1 over root n around beta 0, and we know how to compute the variance. The variance is simply proportional to the inverse of the design matrix and multiplied by the variance of the error. Now, as again is classical in statistics, but maybe not generally to the audience, it's been well known that one of the problems with least squares is that even if, let's say, the errors are symmetric and so on, for example, if they're heavy-tailed, if you really believe, let's say, something like Cauchy, you're in trouble. Remember, you had to have a variance for the errors. And in fact, this old method, least absolute deviations, can handle the Cauchy. And these kinds of methods were studied in a brilliant paper by Peter Huber, quite a while ago by now. Not so much in my view, but quite a while. So you start out with a convex symmetric loss function, for example, the absolute value, or the least squares, and then you look at the estimate, which minimizes, again, the empirical expectation of the deviation. And Huber's theorem is that if C is, which is rho prime, the derivative of this, which is nothing else but the x for least squares, and signum x for least absolute deviations, if rho is weakly smooth, what I mean by that is, obviously, absolute value is not differentiable, but it is weakly differentiable in the suitable sense, so that actually you can make sense of this limit theorem still. And you fix the dimension, so not our usual story. You let n tend to infinity. Then what you get under the same sort of conditions as for least squares is, well, you can go down to the middle here, is essentially the same sort of result. That is, the estimates that you get, which if rho is strictly convex are uniquely defined, asymptotically Gaussian, variance of the order of 1 over, of order 1 over n, and the structure of the variance is the same. You again have the expected value of the inverse of the covariance matrix. All that changes is this, the factor which comes from the scaling. And what that does is exactly what you want. In some sense, and it can even be made precise, what it does is it takes your original errors, which you don't know, and it makes a transformation of these, which can bound them, for example. In the case of least absolute deviations, it basically just this C becomes 1. And so then you get robustness, if you want, that is decent behavior, even in the presence of heavy tails. F is the error density, yes, that's right. And this is, in fact, that's the weak smoothness, precisely. Exactly. Okay, now, as I hope everybody's made clear in their lectures, the current focus of interest is P and N both large. Well, the simplest sort of situation of that kind is when you think that both P and N are large, but P is of much smaller order than N, which essentially can be referred to by this regime. And then Peter Huber long ago, Steve Portnoy later, improved on his results, but more or less showed that life is the same. You still get asymptotic normality, variance proportional to the inverse of the design matrix, or ZZ transpose, and same multiplier on sigma squared. One has to be a little bit careful, and this is already pointing to difficulties that occur as you start to study P going to infinity, is you can ask, well, you're letting P go to infinity, so what's happening to this matrix? Well, one way of thinking about it, which works, it's not the only one, is that you think of the matrix originally as being an operator from Rp to Rp, but that can be embedded in little L2, the space of infinite sequences. And so sigma can go into an operator may or may not go into an operator in little L2. And so you have two things. One is you have the possibility of inference in the same way as before. And also you have what you might expect is that you can estimate the vector as a whole still, even though it's getting to be larger and larger, consistently at least, right? The error of the estimate of the vector suitably normalized goes to 0. Oops. Well, I've already talked about P going to infinity. But what if P is more in the kind of domain that most people have been talking about? That is, the dimension is large, but it's not, and it's not simply of lower order than the sample size. It's, for example, the same order as the sample size, which you can express by, for example, p over n tending to a constant, to a positive constant. Then already Peter Huber in 1973 got a surprising result, which is that in fact there are contrasts which are not Gaussian. If you try to estimate the expected values for the data that you have, then what happens is that the ith observation has too much of an influence on this, and these things, in fact, are non-Gaussian. Unless, in fact, the errors are Gaussian, but in that case, everything is Gaussian. Okay, so implicitly in some way, the way that we've dealt with this is by, I think, is mainly by taking what Hasey, Tibshirani, and Friedman in their lovely book say is the sparsity bet. You use a procedure that does well in sparse problems, since no procedure does well in dense problems. And in regression, sparsity at its simplest simply means that only a small number of factors really matter. All the rest is zero, or in some sense suitably close to that. And then there are a bunch of results. In the sparse case, they're summarized in a very nice book of Peter Budelmans and Sarah van der Heers. More or less, and I'm skipping over technical issues left and right, more or less, if Z1 and ZP are not too correlated in some sense, beta is sparse, as we said before, the signal is strong, somehow that's always needed, and a very weak condition holds that in fact the logarithm of p divided by n goes to 0. In other words, the dimension can in principle grow to 0 exponentially, which is, for example, what one might plausibly think of in genomics and things like that. Then again, as I said, skipping technical conditions, at least if you do L1 regularization, the lasso, and you first, and then you follow by least squares for the non-zeroed out predictors, you basically get an estimate which is as good as if you knew S. And of course, if you knew S, you're back in the old situation. And more or less, although I think I'm probably, this is not necessarily, there may be room for doing this in complete rigor, but more or less the same conclusions have been obtained for least absolute deviations and robust regression. Oops. Oh, this is sort of a side remark, which I can't resist making, is that one has to be careful about the word sparsity, which one throws around a lot. That sparsity is very dependent on the representation. In some sense, that is the real underlying problem. You want to find some representation of the data which is low dimensional. But the trouble is, what representation? Okay, so just to illustrate that point, one of the classic examples, I think Dave Donahoe pushed this to begin with. Non-parametric regression, if you try to take a jump function, express it in a Fourier basis, it certainly has a non-dense representation. On the other hand, if you express it in a Haar basis, sure. And similarly in matrices, which, sparsity in the entries, of course, it can be thought of as in terms of the number of zeros, but then there's also the sparsity having low rank. And those two are not the same. So here's a surprise that I promised you at the end of this. This is work of Nuruddin Al-Karoui, Bin Yu, and Xingwei Lim, and Derek Bean. Really, the essence, the hard work was all done by Nuruddin. And then we had these papers which were first of all heuristic, if you want, and then there was a proof under the assumption that the design matrix was Gaussian by Donahoe and Montanari, and then Nuruddin has in a huge paper shown that in fact these results hold more generally, but not absolutely generally. So you can't do anything about prediction of the vector as a whole, right? If p over n tends to a constant, and you assume nothing about the structure of the vector, you basically you can't do it consistently. But it turns out, both for least squares and robust regression, if you look at contrasts, which is what we're usually interested in, those are still asymptotically Gaussian. However, there's a price. And the price is that this variance is larger than you would expect, and the formula becomes rather complex. And this is all interesting, technical, etc., but let me now show you a simple plot which shows where we started, why we came to this, and why we started to Bin and I started to work with Nuruddin to explain it, because we hit a curious anomaly. And what you have here is the expected value of the squared norm, actually, of estimating beta as a whole for least absolute deviations and for OLS, the ratio of the two. Okay? And you have two kinds of errors: double exponential errors and Gaussian errors. So if you stick to the regime that we've studied so far, which is bounded p, what do you expect? Well, you expect that for Gaussian, of course, least squares does best. For double exponential, least absolute deviations does best. Right? And for Gaussian, least absolute deviations does badly, and conversely. Well, as you'll see, if you look at these curves, the beginning is right. That is, here we're plotting p versus p over n, essentially. The beginning is right, but the curve under Gaussianity is decreasing. The curve for double exponential is increasing, and at some point crosses 1, which is saying that, in fact, you don't have robustness with least absolute deviations, right? Because you have double exponential errors, and least squares is doing better. And this actually, you can say, well, who cares about estimating the parameter as a whole? But this is actually reflecting the behavior for contrasts as well, where you do care. So that was actually the beginning of the puzzle. And if you were interested in the resolution, you might look at or the initial heuristic resolution, you might look at some PNAS papers of ours. Okay. All right. So now, as I said, we were asked oh, how many more minutes do I have? 3, I see. Is that right? Or is it 5? 3 to 5, okay. All right, so we were asked to give challenges. So here is the challenge, if you want, which I've gotten fascinated with. First of all, what is a network? So my definition is, so this is an abstract definition, it's a representation of a set of data objects, could be people, computers, genes, etc., exhibiting relations between pairs of such objects, like friendship, communication, regulation, and usually covariates are present, which are properties of the objects, and properties of the relations. And if you think about it this way, in fact, you can see that networks are an extremely general data structure. You can think of it as a tensor. You have a matrix, and at each ij position, you have the response of the edge, if you want, or the weight of the edge, and this huge vector of covariance. Okay. So I claim that they arise all over. I will just give you, oops, what's happening here? A couple of simple examples. You can see many more. There's a very nice book by Mark Newman, a physicist, called Networks, which sort of really talks about various aspects of the situation as it is now. There are other books as well. So this is a network which consists of political blogs. These are the nodes, and the links are links, one blog being linked to the other. And if you analyze these and use some of the techniques that I'll talk about later, you basically, not surprisingly, find that you can more or less break up this network into two clusters, one corresponding to Democrat blogs and the other one corresponding to Republican blogs, with very little communication between them. I think the yellow is the ones, the ambiguous ones, which communicate both ways. Unfortunately,
Media sites.
Pardon?
Those are media sites.
Is that right?
Yeah.
All of them?
They're yellow ones.
Ah, that's interesting. Okay, I thought that they were simply okay. Well, I guess if you apply, that's true. I don't know, what I'm saying wasn't quite right. If you apply a blocking a method for just splitting the two into two, you have to have two clusters, and then you have to identify the things that so you call everything Democrat or Republican, and then there's the media side. So that's I wonder. Well, I obviously haven't looked at that recently enough. Here's another network which you know, whose status I actually don't know, but in a way, that's what you want, right? It's a network of genes which shows where the clusters have biological meaning, right? But there are a lot of points, as you notice, which are blank. That is, nobody knows what those genes do. They're linked to the others, but we don't know.
How are they linked?
Oh, that's a fairly complicated way. They're used. What's used is, I mean, I'm not vouching for this network, by the way, and methods of evaluating it are unclear. But the links are you have databases of genes with biologically identified functions, and you have patients with or cell lines with certain phenotypes, and you're linking the two in I think I actually well, it's the way is I think described here. Manual examination of the resulting network with the aid of the Gene Mania Cytoscape plugin. Oh hell, no, it doesn't tell you. But that's not all, though. I think is it? I don't think so. I don't think so. That's the thing. It's yes, protein-protein interactions are there, but this is not simply a protein-protein interaction network because, for example, you have identified.
Somatic mutations.
Pardon?
This sentence says somatic mutations.
That's right.
For that gene in the examined protocol.
That's right. That's right. And then further linking these mutations to actual phenotypes, cancer phenotypes. So this is actually is a representation of a whole bunch of data sources put together. in a very complex way. And in a way, the reason that I put it down is it corresponds to in some extent to what we want, but it's a really big challenge for a statistician, right? Because how do you put measures of reliability on these things, right? Some of them are completely undetermined. Some parts have p-values associated with them, but what the p-values mean is not necessarily clear. So there's a lot to be done. Although the final validation is that, if somebody finds a drug which hits all of these, all cancers associated with these genes, you're happy. You don't need to worry about statistical validation of that part. Okay, I'm sorry. I guess I'm running out of time. So types of data, different kinds of networks, questions. Oh dear, that's more than I thought. Okay, so these are a bunch of different kinds of questions that you might ask about networks in general. And a little theory, this is a model, probabilistic model of the simplest kind of network. I should say this. The networks that have actually been studied from a mathematical point of view are absolutely the simplest, namely unlabeled graphs. In other words, no covariates at all. And then for those, just as I forget who, exchangeability was, Mike introduced exchangeability. Well, by exchangeability considerations, you can deduce certain plausible probabilistic models for generating those. And corresponding to those models, there are models which approximate them in a natural way. And these models are these block models, and I'll just describe them quickly. You begin by randomly assigning membership in one of K communities to each node, and then you have a matrix of telling you how to form an edge, and then you just toss a coin with that probability for each edge. And these models, in some very weak sense, approximate. They're like something which would, unfortunately, it's much worse than in the IID case. The IID case, you have an IID sequence of variables. Well, this is like, corresponds roughly to variables which take on only a finite number of values. The analog is IID variables which take on only a finite number of values. But it's much weaker. And then there's some asymptotic theory. All right, and I'll close with this because this is sort of, I find interesting. It turns out that things depend very much on the average degree. I mean, they depend more delicately on other features, but so there are various regimes for asymptotic theory. So the first regime is when you have, again, I'm using the wrong one. When the degree is of order larger than the logarithm of n, then there are procedures which in these block models make no errors, right? So with probability tending to 1, you identify everybody correctly. What's special about this regime? Well, it's also the regime where there are no isolated points in the graphs, because an isolated point you clearly can't identify. Then there's the regime where the degree simply goes to infinity, and you can construct procedures of different kinds with the expected number of errors, the fraction of errors tending to 0. And then there's the regime which there's a huge amount of literature on, which in fact Andrea talked about, which is the regime where the degree is of order 1. And then all sorts of funny things happen. There are phase transitions. You cannot classify individuals with you can't get error rates better than a half for 2 communities in some cases, right? It's no better than random classification. So all sorts of, okay. All right, finally, let me close with the, not just the status of that field. Wait, maybe I do want that. There's lots of work. There are many algorithms and models in the social sciences, computer science, and biology. Some are general, spectral clustering. Some are special. That's one of the interesting things that you find, is that if you look in different corners, you will find ad hoc methods, which are very widely used because somebody has found that they work in some special cases. There are lots of challenges. Stochastic models are rare. Most algorithms are NP-complete. There's a lot of deep mathematical work, but it's largely restricted to unlabeled graphs, and there's not very much asymptotic theory, Bayesian methods. But so I would say this is a real challenge. And with that, I'll close somewhat late.